Hello everyone. Hope you're staying safe and warm on this snow day. I thought I would make this little screencast to capture maybe an abbreviated version of the lecture that we were supposed to have today on Wednesday, January 15th, 2020. Before I begin, just a couple of housekeeping announcements. So first, in case you're not monitoring that discussion board, there's an important announcement about the group projects. So if you go there to the discussion board and look at the issue pages, you'll see an issue about the project groups, and this contains the assignments of each student to project groups. So if you can go there, review your assignments, and on Monday, when we're in person again, plan to find your group and meet up and touch base. Again, if you have any requests to change groups, feel free to actually add a comment to that issue to try to trade things around. All right, and the second sort of housekeeping announcement is that the lecture notes that I'm going to be providing are going to be in the form of HTMLs. So you can open them in your browser. When you click the link on the lecture page, that will actually just open an HTML page, and so you can advance the slides with the arrow button. In case you prefer to look at these or review these in PDF form, you can actually go right now. We might change the link to this, but right now you can find them by going to the website repository and then going to lectures and then lectures 2020, and then you'll see the folder that has these files. So the PDF is under Lecture 4 PDF. And then further, the lectures slides that I'm preparing also involve some plot and some code, and you can actually reproduce these on their own. The source file is in R Markdown, which is also provided in there in case you want to look at see how a particular plot was made, or even run and remake the slides on your own. All right, so getting back to the lecture notes here. So this lecture is a review of probability and statistics. So hopefully you've had a course on interest statistics and probability in the past, but in case that was a while ago, or in case you need some brushing up, this lecture really serves as sort of a launch point before we really dive into the more meaty statistical topics in the upcoming 5 lectures after this one. So this is maybe a good one to have on a snow day, but again, if you feel a little bit fuzzy on these topics, it's really important that you go back and review these and solidify these now so that you can kind of keep pace with the next series of lectures that I'm going to be giving. All right, so for today, as I said, it might be mostly a review. I'm going to go over some philosophy about the field of statistics, go over our goals for this lecture, and review central concepts. So the review of the central concepts is going to include random variables, probability distributions, sampling distributions, estimation and inference, the central limit theorem, as well as hypothesis testing. So we're not going to go very deep into any of these topics, but your goal should be to really be familiar with the terminology and have a very clear understanding of the concepts underlying them. And again, like I mentioned, if you're fuzzy on any of these topics, it's important that you go back and review and really solidify these for the upcoming lectures. So starting at the beginning with perhaps the most basic question, simply, what is statistics? Statistics concerns the science of collecting, analyzing, modeling, and interpreting data, as well as communicating uncertainty about the results. And if you're familiar with the buzzword big data, you might know that the field of statistics is closely intertwined with the fields of data science and machine learning in order to analyze and interpret and really understand these massive data sets that we have in tech and public health and business. And we really want to emphasize that statistical and computational methods should not be used as recipes to blindly follow, and doing so will lead to non-robust science. What we really want to aim for is gaining a rigorous understanding to allow you to perform routine statistical analyses, but also building up a solid foundation that will allow you to follow up on specific and more specialized topics in data analysis. So taking a bird's eye view of the fields of probability and statistics. So specifically, statistical inference is one of the main workhorses for making conclusions about data. So it's a framework for generating conclusions about a population from noisy data from a sample. And so the reason we need to bring in the language of probability is that this enables us to make predictions and discuss uncertainty. And the statistical inference allows us to understand the data and make conclusions. So when we draw our sample from our population, we're learning about the population through the sample and assuming some sort of probability model that governs how that sample behaves, and then using that to learn and make conclusions using inferential statistics to generalize to the population as a whole. So we really need both the probability and the statistics in order to learn from data. So just an overview of the rest of the lecture. So we're going to review terminology and basic concepts. Again, hopefully much of this is review, but if it's fuzzy, please go back and do some extra reading on your own. So we're going to talk about random variables and their distributions. We're going to talk about modeling, estimating parameters, and how the central limit theorem plays a role, as well as the basics of hypothesis testing. So just to define some terms, so a variable is an element, feature, or factor that is liable to vary or change. So in statistical terminology, the variable is actually the unknown quantity that we'd like to study. And in fact, most research questions can be formulated as, for example, what is the relationship between two or more variables? So in an experiment, we might be asking the question, how does a particular variable change when we change conditions? So the condition in that sense would be a second variable. A random variable is a variable whose results from the measurement of a quantity that is subject to random variation. For example, this could be the outcome of an experiment. And other examples of particular experiments would be a simple coin flip or a dice throw, or even the expression level of a particular gene X. And importantly, an important property of random variables is that they have a probability distribution. So just to take a step back before we talk about probability distributions, remind ourselves what is probability. So probability is just a number that's assigned to an outcome or event that describes the extent to which it's likely to occur. So this must satisfy certain rules. So it must be between 0 and 1, for example. But intuitively, what it represents is the long-term frequency of an event. So if an event is very likely to occur, then it has a high long-term frequency. And a probability distribution is a mathematical function that maps outcomes or events to probabilities. So probabilities reflect how likely those outcomes are going to occur. So just to go over an example of these terminologies and add some new ones, we're going to consider this really simple example experiment where we toss 2 coins. So we're going to toss these 2 loonies. Our experiment is tossing the 2 coins. A sample space is simply the set of all possible outcomes. So the set S contains all the possible outcomes of this coin toss experiment. So in this case, we could have 2 tails. We could have the first coin as head, the second coin as tails, or we could have the first coin as tails and the second coin as heads, or we could come up with 2 heads. Sorry, I just realized that these 2 notations here should be reversed, since here we have heads then tails and here we have tails then heads. But nonetheless, these all encompass the 4 different possible outcomes that we could get by tossing these 2 coins. So what could be our random variable of interest? So for example, we're going to talk about the random variable of the number of heads that we obtained from these two coin tosses. So in this column, in this right-hand column, we have the number of heads, and we have 0 heads in the first outcome. We have 1 head in each of the 2 middle outcomes, and we have 2 in the last outcome. So this represents our random variable. Now, there's no reason we have to consider that random variable. We could consider a different random variable. So for example, we could consider the random variable as the outcome of having 2 of the same outcomes on each of our coins. So in that case, instead of number of heads, we would have whether or not the outcome was the same on both coins. So that would be a 1. If we're encoding this as a 1 or 0, there would be a 1 for the first outcome. That would be a 0 for the middle 2, since the outcomes are different on each coin, and that would be a 1 on the last outcome, since the outcomes are the same. But for this example, we're going to go forth with the number of heads. And now, how to assign probability to outcomes. So here I've got the same table, but here I'm replacing with some notation where this little omega represents the outcome. So each of these sets of 2 coins is an outcome. The x of omega represents the random variable. So that's essentially here the counting function that counts the number of heads in each of these outcomes. And we end up again with 0, 1, 1, and 2. Assigning probability of those, so if these are both fair coins, we know that they have equal probability of each of these outcomes occurring, and we have a 0.25 probability for each of these outcomes omega. And notice that these add up to 1, since this set of outcomes encompasses our entire sample space. So there's no probability that any other outcome might occur in this experiment that we don't have listed here. So again, note that each of these possible outcomes is associated with some probability. And we'll also define the term event. So an event is just a set of outcomes that satisfies some condition. And in fact, each realization or each individual value of the random variable corresponds to an event. So to give a concrete example, if x equals 1, so 1 head, that corresponds to 2 outcomes, corresponds to heads, tails, and tails, heads. And so that is an event, that is a set of outcomes that corresponds to the event of x equals 1. So keeping that in mind, we can actually simplify this table from the perspective of the random variable and talk about assigning probability to events instead of just outcomes. So the probability distribution of the random variable x tells us how likely each event or each number of heads is to occur in the experiment. So again, here I'm just collapsing those 2 middle rows, since those correspond to essentially the same value of that random variable, equals 1. And then to assign probabilities to those events, we know that the chance of 2 heads or 2 tails from that previous table was 0.25, and then we also know that there are 2 possible ways, each with probability 0.25, to get x equals 1, so we end up with a 0.5 probability that the random variable is 1. And again, notice that these still add up to 1. They encompass the entire sample space. So this notation here, this px equals x, is the notation that defines the probability function, the probability distribution. And note that you'll see, a little note on notation, you'll see different ways of writing this function. So this is one possible way. Another way is to write this p sub big X of little x, and there might be slight variations even on those ways. And it's not always a p, it might be an f, a little f. So that can cause some confusion sometimes, so just keep in mind that there's different ways of writing that. So in the previous example, in talking about the coin flips, we were talking about a particular type of random variable, and that was a discrete random variable. And that is a random variable that has a countable number of possible values. So this is because we could count up the number of possibilities in the experiment. There were 4 different possibilities of outcomes, and 3 different possible values of the random variable, and we can discretely count those up. There's no in-between values. Other examples of discrete random variables are throwing a dice, genotypes that are measured on a SNP chip, and you could think of many, many other examples. So in contrast, a continuous random variable is one that takes on an interval of numbers, takes on values in an interval. So these can have they may have any possible value within any sort of interval. So examples are expression levels of a gene, blood glucose level, or height of individuals. So there's nothing that constrains these to be in these discrete bins. So talking about continuous random variables, so a particular type of continuous random variable that we're going to talk about a lot in this course are normally distributed random variables. So this is also referred to as Gaussian, and in addition to normal distributions. And the probability density function is given here on the left by this functional form. So that's a function that maps possible values of the random variable x to a probability density given values of these 2 parameters that it depends on. So the mean mu and the standard deviation sigma. So this curve on the right, the blue curve, is this function here. And it's giving us the probability density for any given value of the random variable x. So it does cut off at -3 and 3, but this function continues going, can actually have any real number for random variable x. The probability density function just happens to be infinitely close to 0 as we move out to the tails. And this particular form of the distribution happens to be when the parameter mu is equal to 0. So you see it's centered at x equals 0, as well as the standard deviation of 1, the sigma value. And since these parameters can take on other values, and they might not always be 0 or 1 for convenience, we often write a normal distribution as n of mu and sigma squared. So this is the mu and the sigma squared. Sometimes you might see normal mu sigma, but I think the most common convention is to have the sigma squared. So sigma squared is the variance, and the square root of that is the standard deviation. And again, when we have the particular parameterization of mu equals 0 and sigma equals 1, then this is what's called the standard normal distribution. So again, these parameters don't have to be mu equals 0 and sigma equals 1. Those values can take on other forms to capture different sorts of normal distributions. So this plot is showing different examples where the yellow and red and blue curves all do have mu equals 0. So you can see they're centered on x equals 0, but this green curve actually is shifted over, so its mu value is -2. And in particular, you can see the shapes of these curves are different, and sort of how wide or how narrow they are is governed by that second parameter, the sigma. So the standard normal is the red, and you can see as we decrease the sigma value with the blue curve, it gets narrower, and as we increase it, the curve widens out. And again, all of these functions above are just plot. This is the same probability density function from the previous slide. So these curves are obtained from this function, plugging in those particular mu and sigma values. So a note on relating this back to probability. So before, in the coin toss experiment, we could get a probability from that table by looking at a particular possible value and then pulling that value out of a table. For continuous variables, it's a little more complicated, since we don't talk about the probability that x equals something, since that x value could take on an infinite number of possible values within an interval, we actually need to integrate or find the area under the probability density function to talk about probability. So one thing that's important to note is that the integral, or the area under the entire curve for any probability density function, is going to equal to 1, since that encompasses the entire sample space of possible values. The integral under this normal curve, this is a standard normal curve, integral under that entire curve equals to 1 if we integrate over the entire number line. And if we want to talk about the probability that x, for example, lies between -1 and 1, that corresponds to the area under that curve from x -1 to 1. So keep in mind that that's how we relate the density function to probabilities for continuous variables. So that leads us to statistical inference. So the parameter space is the set of all possible values for the parameter. In the normal case, the parameters are mu and sigma. And one major goal of statistical analysis and statistical inference is to figure out or estimate the parameter values for a particular data set. This is often referred to as fitting the model to the data. So if we have a sample from a population, and we think that it might be normally distributed, a goal here is to estimate those parameters and figure out what exactly is that normal distribution for this particular data set. And this model is a representation that we hope, if our assumptions are correct, approximates the data and, more importantly, approximates the population that the data were sampled from. And once we have these estimates and this model fit, then we can use this to move forward with hypothesis testing to make conclusions about our data, as well as to make predictions about future data or simulations to test model assumptions, for example. So how do we actually go about doing this? So again, the goal is that we are learning about our sample and fitting a probability model to our sample to learn and make inference about the population as a whole. So how do we actually go about doing this? So fitting these models often requires a particular assumption that particular assumption is called IID. And IID stands for, the first I is independent, the second I is identically, and the D is distributed. So independent and identically distributed. So let's unpack that. What does that mean? Identically distributed means that a set of observations are coming from the same population. So in other words, we're assuming that they have the same underlying probability distribution. So for example, the hypothesis test, a t-test, assumes that under the null, all observations are coming from the same underlying normal distribution. So we might not know what that normal distribution is, but doing that t-test requires that we assume that they all come from the same normal distribution. The second I, the first I, independent, this means that all samples must satisfy the condition that the probability of events A and B both occurring is equal to the probability that A occurs multiplied by the probability that B occurs, if A and B are some events. So in other words, the assumption is that the joint probability is the product of the individual event probabilities. And so as I wrote this example statement, this is showing the independence condition for 2 events, but the same definition applies for any number of events. So you could have, for 3 events, probability of A, B, and C all occurring has to be equal to the probability of A times the probability of B times the probability of C if A, B, and C are all independent of one another. So why is this important when we're thinking of fitting a model and performing, for example, a hypothesis test? So in order to assume this assumption, this IID, we have to again assume that our samples are all coming from the same distribution. They're all independent of one another. So one sample's values don't depend on another sample in the same data set, or they're not related in some predictable way. And good experimental design is in part all about kind of trying to avoid that unwanted dependence and violation of independence, because a random experiment, it might be difficult to really assess this unless we start at the beginning with proper experimental design and it basically enforce that the independence assumption is true. So an example of a design that does not satisfy this independence assumption would be if we wanted to measure heights of individuals and we sampled them all from families that were related in some way, and families also that are tall. And if we're trying to generalize that to a population as a whole, outside of that sampled tall related families, this would violate the independence assumption. Since we know that the samples are individuals that are related, they're not independent of one another. So this violates that assumption of IID. All right, so going back to, in particular, the example of the normal distribution and these parameters mu and sigma. So say we have this IID sample, or a sample that we're willing to assume is IID. How do we then go about fitting that model to our data and learning what these mu and sigma parameters actually are? So again, these are, even if we're willing to assume the population or our sample is coming from a normal distribution, we still don't know what the values of mu and sigma are. They're always going to be unknown. So from our sample, we want to estimate them. So this is where parameter estimation comes in. This is our step of fitting the model to the data. So just to define a couple of terms, an estimator is a function or a rule that's used to estimate a parameter of interest. And from that estimator, we can obtain a particular realization or a particular value of the estimator, and that is what is our estimate. So talking specifically about estimators for normally distributed data, so let's say we have a sample of n observations and we're willing to assume that they come from a normally distributed population and that they're IID. How do we then estimate the parameter values for mu and sigma? So recall that the mu is the mean and sigma is the standard deviation of the distribution. So using that knowledge, we can construct estimators to estimate these parameters from our sample of n observations. And so the way we do that is by calculating the average of the values to calculate the mean, to estimate the mean. And we denote that with, since this is an estimate of the parameter mu, we denote that with a mu hat. So that hat distinguishes that mu, which is unknown, the true value, and an estimate that we've actually obtained with values from our data. X bar is just another way to write, an equivalent way to write these functions. It's just summing the values and divided by n. And similarly, the sigma value gives us the estimate, the sample standard deviation. So this formula, summing the squared distances of each observation from the mean and dividing by n-1 and taking the squared root, gives us that sample standard deviation, which then is an estimate of sigma. So then we use that hat to denote that it's an estimate and distinguish it from the true population standard deviation, which is unknown. So given our sample, we have these two estimates for the mean and the standard deviation. And just to drive home the difference between an estimator and a parameter. So in general, remember that the estimator is summarizing the sample that we have at hand. So for example, the sample mean, we're summarizing the sample that we have at hand. The estimator is computed from data in our sample, and we write that with a little hat on whatever the parameter is notated as. So here I'm just using a theta as a generic symbol. And then this is in contrast to parameters, which summarize the population. So these are the ground truth. Parameters are unknown since we don't have the entire population measured. And we write that just with the plain symbol. So in particular, for the normal mean, we have the estimator summarizes the sample in our data set at hand. We estimate that by computing this value from our data set, and we denote that as a mu hat. And for the sample standard deviation, estimator versus parameter, the estimator summarizes sample data. We compute this standard deviation from the sample with this formula here, and we denote that with sigma hat, in contrast to the parameter, which is the ground truth unknown and is denoted with sigma without the hat. So suppose we collected a sample from a population that we, again, assume to be normal, and we estimate that mean just with that formula that we already know with this x bar, and we have this mu hat. Where do we go from there? What do we do with that? And how good is that estimate? So the answer to that depends on a couple of main factors. The first is the sample size. So how many samples do we actually have in our sample? What is the value of that n? The second is the variability of the population. So how are all the samples very similar to each other in that population, or are they all very different? So to talk about the sampling distribution of those quantities that we're estimating from the data, I'm going to first define a statistic, since we haven't done that yet. So a statistic is any quantity that we compute from values in a sample. So for example, the mean is a statistic, since we compute it from data, from values in our sample, as well as the standard deviation. And since our sample values are random, they're a random variable, anything that we compute from those observations, including a statistic, is also random. So because of that, any statistic is also itself a random variable. And because it's a random variable, it also has an associated probability distribution, just as a random variable has, any random variable has a probability distribution. And specifically for the probability distribution of a statistic, we call this a sampling distribution, since we're computing it from a sample. So an example that we're going to talk about in the next several slides is the sampling distribution of the mean. So again, the mean is a statistic. It has an associated distribution. So what do we mean by that? So the sampling distribution of the mean is the distribution of the quantity, the mean that we calculated from data. So the sample mean x bar, again, as we pointed out on the previous slide, is a random variable. So it has itself a probability or sampling distribution. So that's what this figure is showing. So on the left is our population distribution. So it has some mean, some mu that we don't know the true value of. And in this example, that distribution is non-normal. So you can see it has these two modes that decidedly do not look like the bell curve of the normal distribution. But that's fine. We can still take a sample from this distribution. So we're going to pull out a certain number of x's. And from that sample, we're going to compute our statistic, our sample mean, our x bar. And we get a particular value. So say we sampled n x's and we obtained this particular mean here. We're going to put that there. Then we're going to redo that. We're going to repeat that however many times. We're going to sample another random sample of n observations, compute its mean, plot it over here, and so forth until we get actually many different means from many different repeated samples from that population. So that is how we get a sampling distribution. So in practice, we're not actually going to take many samples over and over again, but we know that since our sampling process was random, that if we were to take repeated samples, that we would see some distribution like this. And that distribution will also have some mean. It should have, over the long run, the same mean as the original population distribution. But what can we say about its standard deviation? So this is where the essential limit theorem comes in. So the CLT of the essential limit theorem tells us that the sampling distribution of the mean is normally distributed, and that its mean, the mu of this sampling distribution, mu sub x bar, is actually the same as the mean of the population distribution, and that its standard deviation of that sampling mean is equal to the standard deviation of the population divided by square root of n, where n is the number of samples that we drew in each repeated sample. So this might be a little bit surprising that no matter what this distribution is, even if it's crazy, not normal, that this resulting sampling distribution, that we have some sort of guarantee or theorem that says we know in the long run what that sampling distribution is going to look like. But this does require the assumption that we take large enough samples, which we'll talk about in the next couple of slides. Before that, I'll just highlight that might be a point of confusion, which is the difference between standard deviation and standard error. So recall that from that previous slide, we know that the CLT tells us that the distribution of the statistic x bar, the sampling mean, is normal with population mean as well as sigma squared over n variance, which is the same as sigma over square root n standard deviation. So in terms of terminology here, the standard error of the mean is the standard deviation of this sampling distribution, whereas the standard deviation of x, the population values, is sigma. And so sometimes this terminology in the literature can be mixed up or used incorrectly, so just keep in mind the difference between these two quantities. So just like we estimated mu and sigma before, if we had in a single individual sample, we can also estimate the mean and standard deviation of the sampling distribution of the mean in the same way. So since the mean doesn't change, that estimate is exactly the same. The standard deviation estimate is modified by dividing by the square root of n. So it's the same estimates, except we obtain a different estimate for the standard deviation of the mean, which is the standard error. So the standard error of the mean, again, I mentioned that it might be surprising that we could start out with something that's completely non-normal and end up with something normal for the mean, but it does require a little bit of an assumption. So again, it gives us uncertainty about our estimate of the population mean from our sample, but it does assume that we have a large enough sample. So a rule of thumb is that if the sample size is around 30 or more, that normal distribution is a good approximation for the sampling distribution of the mean. And for smaller sample sizes, if we had a sample of size 5, for example, the standard error is not going to be quite appropriate. It's going to, in general, underestimate the actual uncertainty about our value of the population mean. So, but again, it really does apply, the CLT does apply to any population. We don't have to assume that the underlying population is normal to be able to say something about the distribution of its mean. So for example, if a sample of n could be from a population with a very non-normal distribution, but if we take samples of size n that are sufficiently large, then that sampling distribution, as n grows larger, will approach the normal distribution given here, the sampling distribution of the mean. So that's what this picture example is showing. So we could have these distributions on the top, which are very non-normal. If we take samples of a small n and look at the distribution of the means of those they will start to look like a bell curve, but they're not. They don't look normal quite yet, but then if we increase that sample size to 30 and take repeated samples and graph those do approach a more normal looking curve, as we would expect from the CLT. So this is just kind of a cartoon illustration, but we could do this with actual simulation. So that's what this is showing. So the distribution on the left, this is our population distribution of X. So you can see it's decidedly not normal, centered here at 0.5, but has these very large tails on either end. So if I were to go ahead and randomly sample samples of size 3, I'm going to take 100 samples of size 3 each, and then plot the distribution of the sample means for each of those. So I'm taking the mean of those 3 values 100 times and looking at the distribution of those. So the CLT, if it holds, would tell us that the distribution will follow this normal distribution with this pink curve. So I did that by plotting the mu and this expected standard, or the expected variance of the sampling of the mean. And that's what this pink curve represents. So you can see that in this example with these simulated values, it's got a sort of normal-ish shape, but it doesn't really reflect the variability of the curve. So you can see there's some outliers that make the variability and show that standard error really is an underestimate. So I can increase that value of the sample size, the same exact setting, except I'm taking 100 samples of size 10 and plotting the distributions of those 100 means. Looks a little bit more normal, at least there's only one mode, but there's still a little bit extra variability maybe. And then I can increase that to 30 or even 100 and show that just really gets tighter and tighter, matching that normal curve, showing that estimate for the standard error of the mean does become appropriate and very accurate, even starting with this non-normal distribution. All right, so now let's move into hypothesis testing. So just to define a few terms. So a hypothesis is a testable idea for explaining a phenomenon. So testable means that we can falsify it. A statistical hypothesis is a hypothesis that is testable on the basis of observing a process that is modeled via a set of random variables. Hypothesis testing is any formal procedure for determining whether to accept or reject a statistical hypothesis. And carrying out this procedure requires comparing two hypotheses. So one is the null hypothesis. This is what we start out assuming. And this is denoted with an H sub 0, and we often say H naught to denote null hypothesis. The other is the alternative hypothesis, and we often write that as HA or sometimes H1 in contrast to H0. So H naught or null hypothesis versus HA. And what we are doing in the hypothesis test is determining whether or not we accept this null, which we start out assuming, or reject and say we falsify this null, therefore we have evidence to suggest that the null is not true, which we're left with the alternative hypothesis. So a motivating example of why we might want to carry out a hypothesis test, so let's consider the expression level of particular gene G that we measure in N patients with disease. So we have these Z1 to Zn, those are measurements of gene G in N disease patients. And then we have M control or healthy samples. So we have Y1 to Ym healthy samples, measurements of that gene. And we want to ask, is gene G differentially expressed in our cancer samples compared to our control samples? So here the null hypothesis, if we're looking for a difference in that expression, is to look for whether the means are the same in the cancer versus the control. So our null, we start out assuming that they are the same. Our alternative is that these two population means are different. So in our test, we want to determine, do we have evidence to falsify this null that they're the same? And the hypothesis test allows us to determine, in general, whether the observed differences that we observe in our sample are significant or not. So the basic steps in any hypothesis test are first to write down the scientific hypothesis as a statistical hypothesis. So putting this in terms of parameters that we can estimate and construct a test statistic. Step 2 is to actually define the test statistic or the random variable that corresponds to the question. And you typically know or formulate this so that you know the expected distribution of the test under the null. So you assume that if that null hypothesis is true, that your statistic is going to follow a certain distribution. And then in step 3, you compute the p-value, which is associated with the observed test statistic, by looking at the likelihood of that observing if the null were true. So if your p-value is extreme, as if it's extremely small, that means you were very unlikely to observe that given the null hypothesis, and that would be evidence to reject that null hypothesis. So going back to that motivating example of cancer versus healthy gene expression and determining what factors determine whether you're likely to get a significant result or not. So this plot here is showing a sample of 4 cancer patients and 4 healthy patients and their gene expression value, some normalized gene expression value. So that would be a sample of 4. So generating from the same model, I simulated 8 samples from the cancer and the healthy group. You can see there still doesn't look to be much difference between the 2 samples, but if I sample a larger sample, say of 20, then you can start to see that there might be a difference in that the cancer samples have higher gene expression for this particular gene than the healthy samples. So in this toy example, I drew all 3 of these samples from IID, so independent and identically distributed normal distributions that have equal variance, and they also have a difference in means between the cancer and the healthy groups by 1. So actually the mean of the cancer groups was set at 1 and the healthy was set at 0. So you can see that even though that difference persisted, the underlying difference persisted for all 3 of these groups, you can really only start to really have evidence for it in the last plot. So what does this mean? This points out that sample size has a big influence on whether or not you're going to be able to determine if there's a significant difference between, for example, 2 means. So this is showing kind of that same paradigm in the top row here, where I've got samples of size 4, 8, and then 20, but all with the same standard deviations of 1. And then I'm also adding what the distributions look like if we have a larger variance or larger standard deviation of size 3. So this grid kind of explores for the same difference in means. So here each of the pink curves are centered at 0. Each of the blue curves are centered at 1. So there's always a mean difference of 1. But you can see that those curves look more separated or less separated depending on both the sample size and the spread or the standard deviation. So you can see it'd be harder to detect a difference between these 2 distributions with a small sample size as well as a large standard deviation compared to these 2 curves, which look very well separated for a larger sample size and small standard deviation. So it's really both of these factors. They each contribute to how likely we are to be able to reject the null hypothesis if the alternative is actually true. So a particular hypothesis test that is perhaps the most common hypothesis test for continuous values that I'll go over is the t-statistic, the t-test, which involves using the t-statistic. So this is a test that can be used to measure differences in means that are adjusted for the spread or standard deviation. So in particular, you can see that in the form of the test statistic. So you can see the numerator captures that difference in the sample means, and then in the denominator adjusts for the spread or the standard deviation. So going back to this slide, we want to look how separated are these compared to how wide each of these curves are. So that's what the denominator is measuring on the t-statistic. In particular, to estimate the standard error of the difference in means, we use this equation here on the bottom. So this is a little different than the equation you saw before for the standard error of one population or one sample mean. So the difference has to take into account the sample sizes of two different samples. So that's why you see the square root of 1 over n plus 1 over m term. And then also accounting for the fact that we have two different samples. So we have to calculate the sample standard deviation for both of the different samples. And that's what this SP is, stands for pooled variance here in this equation. So putting that together, that gives us the denominator of this test statistic. So in order to obtain a p-value and know whether or not we're going to reject our null hypothesis based on that test statistic, we need to know the distribution of that test statistic. And from the theory, we can get that distribution under some set of assumptions. So for example, we're going to talk about the equal variance t-test. So if we assume that both populations we're drawing from, the normal and the cancer in our example, have equal variance, as well as the normality of those two populations, then we know that our t-statistic that is given by this formula here follows a t-distribution with degrees of freedom m plus n minus 2. So there's actually an alternate form of this t-test where we don't have to assume equal variance. It has a different variance estimator that we'll talk about in an upcoming lecture. But this is the equal variance version. So given that, once we have our test statistic and its corresponding distribution, we can see how extreme our sample is and obtain our p-value. So the t-statistic, the value of that tells us how extreme our data is compared to the null distribution. And we actually get that p-value by computing area. So this is getting a probability essentially to the left and or right of the t-statistic that we observe. So for example, if we observed a t-statistic of 2 and we had 5 degrees of freedom, we would be looking at, for the null hypothesis of mean 1 equaling the mean 2, we would look at the area to the right of 2 on the blue curve and add that to the area to the left of 2. And then we would only look at one of those tails if our null hypothesis was one-sided. So if we were testing whether the population mean from one sample, one population was less than the other. So to wrap up this lecture, we covered some basic concepts in probability and statistics. And in particular, we learned that random variables are variables that have an associated probability distribution, and that any statistic we compute from sample data is also itself a random variable and has its own probability distribution. And the central limit theorem, or CLT, gives us the sampling distribution, the form of the sampling distribution of the mean for any population. Does not have to be normal, as long as our sample size is high enough. And finally, hypothesis testing gives us a framework to assess a statistical hypothesis under the null and tell us whether we have evidence to overturn that null hypothesis. And we went over a particular example of a hypothesis test called the t-test. So we will pick up where we left off on Monday in person. And again, if any of these concepts don't seem quite solidified, it would be great to review those so that you're ready to go starting on Monday.